And our goal of this trial, it was an add-on to another trial, was to evaluate the effect of this vaccine. It's a live Salmonella vaccine developed at Iowa State University with Drs. Ted Kramer and Mike Roof, to look at the post-harvest Salmonella contamination of pig carcasses. And we looked at it in two different fashions that we'll talk about in the study. The trial design was set up to start with pigs on the farrowing site. So we wanted to give the vaccine a maximum challenge by having to protect the pig all the way through its life cycle to market. So we had litters of pigs in the farrowing crate, and they were blocked by farrowing date or age of the pig and parity of the dam. So that was the same amongst the two treatments. Litters were randomized to treatment, and that was applied 7 days before weaning. And piglets were either drenched with the vaccine or received nothing. And we did it at this time frame to do two things: to allow the pigs to have some protection already early in the nursery. Dr. Schwartz talked about timing. This vaccine takes between right around 2 weeks to mount a fully protective immune response. So if we did it before weaning, we know early in the nursery that pig's going to have an active immune response and be protected. So that was part one. Part two, this is in a virulent live culture. It's in a virulent live vaccine. It can be shed. So if I would give it to pigs in this litter, mix them right away with other pigs, potentially it could shed from the vaccinates to the non-vaccinates and then interfere with the results of the study. So the pig was the experimental unit. An entire litter of pigs was vaccinated or not. And really from that point in time forward, everybody was blinded as to who was who. We had a piglet number, but every pig had a unique number. We didn't know what treatment they were. Ear tags were used. They were the same color for both groups. So really from the point in time where they got the vaccine forward, everything we did was blinded. So until the very end of the study, nobody really knew what pig was what, which is important statistically and for biases. We didn't allow any movement before weaning, again, so we couldn't shed between groups. At weaning, those pigs were put onto a common transport and taken to a wean-to-finish barn. Non-vaccinates were up above, vaccinates on the bottom, just in case the vaccinated pigs would be shedding any at all. We didn't want them to cross-contaminate. Treatment groups were randomly then mixed within a pen. So from the time they got to the finishing barn in Iowa forward, they were commingled. So feed was the same, pens were balanced, etc., etc. From that point forward, there was no difference between the groups, just to allow for all of those factors that influence Salmonella to be the same among both pigs. During the study, we collected serum samples, oral fluid samples, and fecal samples from within the pens at designated intervals. What we found was Salmonella infection was very widespread. We had about 90% seroconversion by the end of the study. So a large number of pigs had been exposed to some kind of Salmonella. At harvest, we selected 100 pigs per treatment randomly by the ear tag number, identified them, had about 80, 85 of each group, sent them to a packing plant. What's important to note, this infection was widespread, but if you go back to that chart that Dr. Schwartz had, typhimurium is the main cause of clinical disease. In this barn, we did not have any clinical disease. We couldn't identify any pigs, any episodes, anything that would look like an enteric infection with Salmonella. So in the live animal phase, there was no economic damage to the controls, hence the vaccines had no opportunity to show a benefit. We said, let's take them to the packing plant and look at the food safety side of things. So they were loaded and they were transported 3 hours to a small packing plant in Sioux Center called Supreme. And I want to say thank you to the folks there because they were wonderful in how we did this process. They were loaded into separate compartments of a cleaned and disinfected trailer. So that again was blocked to control. Both groups were the same. They were unloaded and put into adjacent, washed, and disinfected pens at the plant. And they sat there overnight. Both groups were slaughtered during the first shift in the morning, so the plant would be clean, cross-contamination risk would be minimized, etc. We used the Specie sponges just as a way to collect various samples. And what we did was we swabbed the trailer they came in on, the pens at the plant, On the production line, the peritoneal cavity of each individual pig was swabbed. We pulled the entire viscera set off then and collected the ileocholic lymph nodes from each individual pig. The carcasses went into the chiller. We came back the next morning and swabbed using the standard USDA process at the jowl, midline, and tail head. So we wanted to compare both the standard USDA process as well as what might be inside the carcass by using these lymph nodes. All samples immediately went to the Iowa State University Veterinary Diagnostic Lab. They were enriched and processed. Dr. Frana from the VDL reported on this at AASB last year. The positive cultures went to the National Veterinary Services Laboratory to be identified. And there's two very cold veterinarians after about 3 hours in the cooler wiping pigs. The take home. In the vaccinated group, we did not identify any salmonella from any of the lymph nodes. Perfectly clean. From the unvaccinated pigs, we had 5 positive lymph nodes out of the 78, or about 8%. And Iowa State University did the statistical tests on this, and that was a statistically significant reduction in salmonella. This just repeats the results we've seen in other studies. It's not always a 0 in vaccinated pigs, but it's always significantly less. And that really is our take home. There's probably going to be some salmonella in those lymph nodes of unvaccinated pigs. If that's a risk, we know we can reduce that risk because we've repeatedly shown the ability of this vaccine to do that. The conclusion then of the study, SC54 vaccination significantly reduced the post-harvest contamination in this infected barn and transport. So therefore, vaccine could be both a tool to help the pig clinically, but also a tool to help enhance pork safety. Less salmonella in those lymph nodes, less salmonella in the carcass. As I said, this is kind of a repetition. It's one of multiple studies. We've looked at various studies throughout more than a 10-year period of using this vaccine to repeat the same thing. Sometimes we did whole barns vaccinated, whole barns controls, etc., but always that same result. And that's been strong enough that the USDA has allowed us to put a package insert approval documenting that this vaccine will reduce salmonella in the carcass.